Hello, my name is Francisco Castro, and today I'm going to be taking you through my summer research project, which I did at the University of New Hampshire under the guidance of Caitlin Van Volkum. My research title is Slipper Limpet Population Increase and its Effect on Local Predators. The slipper limpet, or Cudipilla fornicata as it's known scientifically, it's a type of marine mollusk. It lives in stacks comprised of different individuals. The ones at the bottom are always female, while the ones at the top are always male. They are hermaphrodites, meaning they change sex when necessary, and in this case, it's when the oldest female dies, the oldest male takes her place as female. They are suspension feeders, meaning they filter nutrients and plankton from the water. In northeastern USA, which is where they're native, they reside year-round, and they can live up to 8 to 10 years in the wild. So, a little background as into why we're interested in this organism. There's been a historical increase in their populations over the years. They have been shown to be very resistant to climate change and changes in their environment. They have a very wide diet, and what this means is they can filter a very wide range of sizes of nutrients and particles from the water. And they thrive in warmer water, which would be a problem if for some reason the oceans started warming up. Oh wait, climate change. Now, what have been the tools that make us think they're increasing? Well, I'm going to cover what local observations are, that there has been an increase in their reproduction period over recent years, and what we call fouling panels. So, local observations include recreational divers, but mostly local scientists that are doing censuses and keeping track of the population of these organisms in different areas. So here I'm presenting a graph that was done by Caitlin Van Volkum on density of limpet stacks per square meter over 4 different states in northeastern USA. Here in the yellow you have Rhode Island, and the purple Massachusetts, and the green New Hampshire, and then Maine in the darker blue. So as you can see over this scale in the y-axis, in some cases you have over 900 limpet stacks per square meter in the substrate, which is an insane amount considering a square meter isn't that big. And that is fairly consistent over the 600 to 500 limpet stacks per square meter range over all the states. And so the other method that scientists are using to identify populations increase in these limpets are a thing called fouling panels. And fouling panels are just pieces of glass that are put underwater in a site and left there for a number of years or extended period of time to record what colonizes in them. And as you can see, that's marked over here. In 1985, we only tracked 2 limpets that colonized these fouling panel. But then jump forward in time to 2019, and you have over 30 limpets that are colonizing these fouling panels. And this is fairly consistent over the sites and over the years. And in each and every site, there's been an increase in population, or an increase in colonization rate, I should say, of limpets throughout the years. So, why do we even care if they're increasing in population? What does this mean for the ecosystem? Well, we'll have to cover 3 things: organic waste, less physical space for other species, and effects on predators. And just to get this out of the way, less physical space for other species is just that. They're taking up so much space, their population is increasing so much, that they're leaving little to no space for other native species to thrive. So, touching the point on organic waste, the slipper limpet as an invader in places such as France and sometimes even in western US has been shown to form a thick layer of anoxic mud. The organic waste can come in the form of them dying or in then excreting a lot of waste as they digest their food. But this layer of anoxic mud actually removes oxygen from the sediment, killing everything that needs oxygen to live down in the benthic zone. And, of course, the highlight of this investigation is the effects on its predators. While their increase in population could mean that preferred food items for the predators are now scarce, it could also mean that they now have a limited choice in prey selection. This could possibly impact predator behavior, and it could possibly impact their nutrition. And to better understand the effects of slipper limpet population increase on predators, I conducted what is known as a prey preference experiment. And how I did this was I chose a location, which in our case is Nahant, Massachusetts, and I chose it because, in theory, we know that the predator that I chose interacts naturally with the slipper limpet. And the predator that I chose is none other than the Asian short crab. And the Asian short crab is a very small species of crab, scientifically known as Hemigrapsis sanguineus, that is considered to be an invasive species in northeastern USA. But because it's been an invasive species for so long, a little more than 40 years, it's considered to be a local natural predator for the slipper limpet. And how the experimental setup for this experiment worked is I isolated each predator in a tank for it to be a controlled environment with 24 total prey choices. And those 24 choices consisted of two different species of prey items. One of them was Credipula fornicata, which is the slipper limpet we've been talking about, and the other one is the blue mussel, which is another native species of marine mollusk that the crab, in theory, could choose to eat. And so I had 4 different side classes in each of the tanks for a total of 24 prey items. So I had an extra small, a small, a medium, and large, and 4 of those for each of the prey items. In this image, you can see how we kept the mollusks and the slipper limpets from actually moving, because although they're considered to be sedentary animals and they don't move much their whole lives, when they're faced with a threat, such as a predator, they can move, and they can move fairly well. So in order to protect the experiment from being biased or hindering the results, we decided to glue them to ceramic tiles with superglue and place them at the bottoms of the tank. And the timestamp for this experiment was a total of 24 hours for each individual crab in the tanks, and we had a total of 43 crabs go through this experiment. So as you can see here in this image, this is our experimental setup. We had tanks that were about 15 gallons each, and each tank had one of the crabs and all 24 food items dispersed unevenly so that it wasn't biased or anything. And we left them for 24 hours. We only had about 7 tanks, so we had to repeat this experiment a couple times to reach that 43 mark. So, after running the experiment for about a week and a half and analyzing the data, we finally had our results. And they were quite shocking, honestly. So, this is a graph that shows number consumed versus the prey species, the slipper limpet being on the left and the blue mussel being on the right. As you can clearly see, the extra small sizes were favored for the crabs versus the large sizes that weren't even touched by the crabs. And this could be justified because the crab is a smaller crab, and maybe the smaller sizes are the ones that are most accessible to it without expending that much energy. But if you quickly look at the data, you can analyze that there's more bars for the slipper limpet, which leaves us to imply that they actually prefer to eat the slipper limpet over the blue mussel. And if you see this graph, on average, the slipper limpet on the left was eaten or predated upon more than the blue mussel. Now, what does this mean for the crabs, that they're preferring to eat more of the slipper limpet over the blue mussel? So, to better understand the implications and nutritional value that these crabs might or might not be getting from eating the mussel over the limpet or the limpet over the mussel, an hepatopancreas to body weight ratio experiment was conducted. And so how this experiment worked is the crabs were fed either a strict diet of limpet or a strict diet of blue mussel, and then over the course of a few weeks, their hepatopancreas was measured in relation to their body weight. And so a heavier hepatopancreas usually means a healthier crab. And as you can see, in all of these cases, in this 4 species of crab, the hepatopancreas was a lot heavier, or fairly heavier, for the ones that were eating the blue mussel. In contrast, the hepatopancreas to body weight ratio was lower when the crabs were fed the limpets, which implies that they are getting less nutritional value when they're choosing to eat the limpets over the blue mussel, which is what we saw in our experiment, that they chose to eat the limpet over the mussel. So, at least in this 4 species of crabs, the Emigrapsis is not included here because of time implications, but I got back to my mentor, and she did say that it was the same result for Emigrapsis. They are receiving less nutritional value from the things that they are deciding to eat. And another area of my research that needs highlighting is shell morphology. Now, shell morphology can vary depending on geographical region, temperature of water, type of substrate that they're growing on, and it can actually impact how likely limpet is preyed upon by a crab. And so what I did is, over the course of a few weeks, I measured different shells from different locations, all the way from Maine to Rhode Island, a lot of different points, over 20 points, and I found out that shell thickness is actually the most important aspect of limpet being preyed upon. So as you can see in this image, here circled in red, this is the area that crabs, lobsters, and all the predators of the limpet go for when they're gonna eat a limpet. This is because it is known that this is a weak spot in their shell and it's easier to break. Now, depending on shell morphology, this area can be thicker or thinner, which can impact how likely the limpet is preyed upon. And I'm gonna show you this graph of shell thickness over different sites in 3 states, Massachusetts, Maine, and Rhode Island. And as you can see here, it varies a lot. Shell thickness can vary a lot, especially on the age, on the substrate, on the temperature of the water. There's a lot of variables that go in it. But in average, shell thickness is higher in areas that are warmer and that have a rougher substrate for them to grow on, which means that not only are they facilitated to grow in warmer waters, but because they're thicker in shell, they're less likely to be preyed upon, increasing these populations exponentially. And so, in conclusion, we could expect that as climate change goes on and water temperature keeps rising throughout oceans in the world, the populations of slipper limpet will just continue to increase. And that increase in population means that it would impact the ecology in ways that we've yet to understand. But what we can theorize from what we know and the pre-experiment that we did is that predators may need to change their behavior to compensate for that nutritional loss, because as we stated, the ones that are eating more limpets tend to be less healthy. And the other aspect, and the last aspect, is that different snail morphologies could have different effects on their environment, not only on how likely that snail is preyed upon, but ongoing research suggests that filtration rates and organic waste rates also are impacted by the shell morphology. So in my acknowledgements, I have to mention the people that were indispensable for this project. Up first, Dr. Jen Dykstra. Without her knowledge, her guidance, her mentorship, this would have been really difficult or maybe impossible. Of course, Caitlin Van Volkom. Without her research data, she basically took me under her wing. She was indispensable for this research. Of course, Dr. Nancy Kinner and Kathy Mansager for creating the program and accepting me in it. And last but not least, the University of New Hampshire for having me, accepting me, and letting me use the facilities and the resources necessary for this experiment. Here are the references and the resources I used for building this presentation. And of course, I have to acknowledge the people that weren't directly associated with my research, but did help me get through the internship, which are my friends and my fellow interns. Thank you all for watching this video. I hope you liked it, and I hope you have a good rest of your day.